to. Our next speaker, Lea Bartsch, is currently working at MGH, Harvard Medical, and she's working in the lab of Georg Lauer, who ProImmune has had quite a bit of kind of history with, working over the years. All right, so like I said, Lea is working at MGH, Harvard Medical with Dr. Georg Lauer. Her research interests are in T-cell immunology in chronic liver disease, with a particular focus on T-cell dysfunction in chronic viral infection. She completed her medical school in Lübeck and Hamburg in Germany. During her doctoral thesis, she investigated Th17 and Treg balance in the context of chronic inflammation and hypoxia. Before she started to work at the MGH, she began her residency in the Clinic of Infectious Disease and Internal Medicine in Lübeck.
Thank you so much, Andrew, for the kind introduction. Yes, I would like to share our approach to use tetramers and pentamers in HPV infection. And rather than presenting one project, I just want to present our challenges with pentamer and tetramer staining. The first of all, a short introduction about HPV infection. So HPV infection is still a worldwide problem. Although a vaccine is available since decades, we have around 250 million chronically infected patients globally. And HPV is still responsible for over 800,000 deaths per year. And as you can see on this map, it is especially a problem in Africa and in Asia and South America. So in countries where the vaccine is not available or the treatment access is limited. The long-term complications of a chronic HPV infection is liver cirrhosis, liver failure, the development of hepatocellular carcinoma, or in the worst case, in a chronic infection, 25% of these patients die. I don't want to go into much detail about the virus and the viral structure. I just want to highlight some specific characteristics which are important to understand the talk. So HPV is a DNA virus and consists of different proteins which can be recognized by T cells, the polymerase, the core, the X, and the S protein. And when HPV virus enters the hepatocytes, it can persist within these hepatocytes by forming a cccDNA, a persistent form. And importantly, this form cannot be reached by our current treatment strategies. So it's really important to improve them. Another important characteristic of the virus is that the age of infection determines the outcome. So when an infection occurs in infants, like it is in high endemic country like Africa, most of the infants cannot control the virus, and this infection leads into a chronic infection, and this is accompanied by HBs antigen persistence. Whereas when we have an infection in adults, 95% of them can control the virus and build up an effective immune response. So for us, it's really interesting to understand the immune response or the differences of the immune response which occurs in infants in comparison to adult patients. When we look at the cause of a chronic infection, this can be divided in different stages, and these are divided by the HPV DNA level, which is or represents, indicates the viral replication, and the ALT level, which replicates which indicates the liver damage, and the liver fibrosis. And the end of the aim of a treatment of a patient is functional cure, where the HPV DNA is not detectable anymore and the ALT level is normal. Important to know is that just in under 1% of chronic HPV patients, this goal can actually be reached. And another important a characteristicum or finding is that the cccDNA is still present in these patients. So why are T cells important for HPV infection? So during chronic infection or during cancer, T cells, there's a persistent antigen presentation for the T cells, and this can lead to a stage of T cell exhaustion, which is accompanied by a change in the metabolic profile of these T cells, by decreased cytokine production, and overall this leads to an ineffective viral clearance. An important question for us and for others is whether this stage of T cell exhaustion can be reversed, for example, by treating the patients. So chronic stimulation, antigen stimulation in HPV leads to this exhaustion phenotype, and there are different exhaustion phenotypes pre-existing, and this depends on the viral load and also on the disease stage the patient is in, and it can also vary between different epitope specificities. Overall, very little ex vivo data is available for HPV, for the function of HPV-specific T cells, and this is even fewer data is available when we look in the liver. So overall, a better understanding of the immune response in HPV infection is important to improve the treatment strategies of our patients. Our aim, or the starting point, is that we want to understand these changes and that there is very limited, so the epitopes which are described as are very limited, and the cells comparable to the other talks have a very low frequency, and we have a limited access to liver tissue in humans. Our aims are to create an epitope library to generate new tetramers, and this we do in cooperation with ProImmune. And then we want to identify these cells directly ex vivo and characterize and phenotype them. So the first approach to create or expand our repertoire is that we use a ELISpot approach. We generated a peptide library covering the whole HPV DNA, and then stimulate acute and chronic infected HPV patients with the peptide pool and single peptides, and then we're able to potentially detect the interference signal, and this interference signal can then be confirmed by using intracellular cytokine staining of the same patients. And with the intracellular cytokine staining, we are not just able to analyze or detect the interference signal, but also distinguish between CD4, CD8 response, and for sure analyze more than just cytokine expression and additional activation markers. When we then hopefully, in the best case, find the right or the optimal peptide by using different experimental approach and also databases to calculate these optimal epitope, we then create a tetramer, or this actually ProImmune does this for us. This is an example for a custom-made tetramer or pentamer, and then we validate these new epitope in a pentamer staining. Here I show an example of two acute infected HPV patients where we performed the intracellular cytokine staining, and as you can see, we are able to detect a CD8 and also a CD4 response, and in 80% of our acute patients, we could find these responses, and in 50%, around 56% in the of the chronic infected patients. So that's really hard to detect them in the chronic infected patients. Here on this slide, I show an overview of all the responses we could find in our cohort for the CD8 T cells and the CD4 T cells. On the right-hand side, as you see in the acute infection, the responses cover the whole genome of HPV infection. But as you can also see in the CD4 T cells, we have fewer polymerase responses in comparison to the CD8 T cells. And when we look in chronic infection, the responses are very limited. After we now designed some new tetramers. We now for sure want to detect our pentamer and tetramer positive cells. Here I show an example of a tetramer staining. So this is a CD4 T cell response in acute, chronic, and seroconverted patients. In the acute patient, we can detect in 80% of the patients with the matching HLA type. We can detect a tetramer cloud, and we do not need to further enrich these tetramer clouds. But in chronic and functional cure patient, we always use magnetic enrichment in order to see these tetramer positive cells. And in the chronic infected patient, we can just detect them in 20% of the patients. So it's really hard to detect them. When we then look in the CD8 T cell compartment, and here's also an overview across the different disease stages, you can also see that the frequency is very, very low, but we are able to detect them in all the different stages. And one approach, beside to the magnetic enrichment, is for us to enrich these signals is to use cell lines. So we always stimulate the patients with the corresponding peptides and incubate them for 12 to 14 days in order to detect a huge tetramer cloud. But for sure, when we do this in vitro incubation, this also changes the phenotype of the cells. And what we originally want is to really characterize these cells directly ex vivo, but the cell lines just help us to screen the patients and to really know what's happening. So for the phenotyping of the T cells in the different disease stages, we have different approaches. The first approach is to perform a FACS staining. And here's an example of FACS experiment, so where we actually stimulated CD4 T cells with the corresponding peptide, and then measured the cytokine expression. And here we measured interferon, TNF, and IL-2. And when we measured the cytokine expression in CD4 T cells, we cannot combine this with a tetramer, but this is possible for CD8 T cells, because when we do the combination, the T cells loses their tetramer signal. So we need to add additional markers like CD40L as an activation marker. And what you can see here is that in the acute patients, the CD4 T cells really produce a lot of cytokines, and this is a little bit reduced in the functional cure patients, but in the chronic HPV patients, there's really a lack of cytokine production. In addition to the cytokine expression, we for sure can also measure surface molecules, but also intracellular markers with the FACS staining. And here's an example of a PD-1 staining where we measured or compared the differences of PD-1 expression between different epitopes. And PD-1, as you all know, is an important activation marker of T cells, but also a marker for T cell exhaustion. The problem when we analyze these cells by flow cytometry, in a flow cytometry experiment, you always need to know what you want to look at, and also the markers are limited. So therefore, we have an additional approach where we use single cell RNA-seq in order to really comprehensively phenotype the virus-specific cells in the blood and also in the liver. And for the liver samples, we use fine needle aspirations of the liver because this is easier for us to get in comparison to a liver biopsy, as it's minimal invasive, there are less complications, and it's easier to repeat. And why do we need to look at the liver? For sure, we are interested at the site of infection, and we know that the liver itself can change the phenotype of T cells, and we really want to know what is happening in the liver in comparison to the blood. So what we do is we analyze, so we single sort the FNA and blood samples, and for sure we do this directly ex vivo, not with a cell line. And then we perform a SmartSeq experiment to a SmartSeq analysis to comprehensively phenotype these cells. Here I show an example of a sorting experiment of a fine needle aspiration of the liver, where you can see the tetramer cloud, and often the percentage of tetramer positive cells within the liver is higher than the blood, than in the blood, because of the different composition of or percentage of the CD8, CD4 cells in the liver, and also because the cells are enriched at site of infection. What we can analyze then, and the first step, like always in the single-cell RNA-seq, is the clustering of these cells. And as you can see, we within a, and these are just specific T cells from acute patient which who cured the virus. You can see that there are different clusters or different population within the specific T cell cluster. When we then look at the genes which define these different clusters, it is interesting to see that there are different stages of T cell activation within the specific T cells. So we have a polyfunctional T cell stage, a memory-like phenotype, and the intermediate stage of T cell activation. For sure, it's also possible to compare the different epitopes. And here we looked at the gene expression level between the different epitopes and important markers like granzymes and granulocytes within our population. And we saw the biggest difference between the polymerase and the core-specific T cells. And this was also the case when we then looked at different pathways which are enriched or activated in these cells. And for example, when we look at T cell activation in comparison to the PD-1 activation. So this should just give an example of what we can do when we use the SmartSeq approach, because then we really can look at many different markers in comparison to the FACS experiment, where we really need to know what we want to look at, which is also for sure important, but the SmartSeq approach gives us more opportunity to look at different markers. So to sum this up, it is still necessary to expand the tetramer repertoire, and we really want to investigate the immune response at site of infection, and we want to comprehensively phenotype these virus-specific cells and the challenges that the frequency of them is very low. And the overall goal for sure is to improve these treatment strategies for the HPV-infected patients. With this, I would like to thank all the people who are involved in this project, and as Andrew said in the beginning, I'm part of Georg's Lauer Lab at MGH. And with this, I would like to thank you for your attention. Thank you.
Thank you for that. So we'd be happy to take any questions for Lea. I know I had a question. I just wanted to know if you had any thoughts or what your group's current thoughts were about why in the chronic infections, a lot of the epitopes that you're seeing are directed towards the envelope region, whereas in the for the acute infections, they're a bit more kind of all over the place. I know there's a lot more of them also, but.
Yeah, we don't know yet, but when we look in the SmartSeq data, it looks that the envelope specific cells have really a different activation and exhaustion phenotype in comparison to the polymerase specific ones. So the polymerase specific ones are the ones who are really activated and can produce the cytokines in comparison to the envelope ones which seems to be a little bit more exhausted. So it could be that the chronic patient really lack the polymerase specific ones, and this might be one of the reasons why they are chronic. But we are still in the process to analyze our SmartSeq data. So I don't have a final answer for this, but we hope to find this. And this is really one of our interesting findings findings that these different specificities have different function within the HPV infection.
Yeah. Thank you.
Thank you.
Emily, did you also have a question?
Yeah, it's just actually just a simple, easy technical question. And you may have mentioned this, and I'm sorry if I missed it, but for your ELISpot analysis, because the precursor frequency of your T cells is so low, are you doing an overnight, or are you doing like an extended, a week long?
No, overnight.
If you're doing overnight, have you tried doing any extended co-cultures like DC enhanced ELISpots?
So I personally didn't, but I know that Ruben, the postdoc before me, did some experiment and for what's the best approach to actually detect these cells. And in the end, we decided the overnight is the best for our cohort. Yeah.
I have a question now.
Okay, Shivali?
Yeah. So I just wanted to ask you if you had different cohorts of HBV infection, right? Did you try occult Hep B patients? And like if you tried, do they behave similar to the recovered ones? And my second question was, did you try grouping your patients according to their e antigen status?
Yes, great question. So yes, we did this. We have a very, I didn't show the data today, but we have a very great cohort where we have patients in different immune stages. One, I mean, all different stages with E antigen present and also the S negative ones. And we have blood and liver samples of them and sorted many of these patients. And we are still in the process to analyze all the data we are generating with the SmartSeq approach. So this is the answer to you. I didn't understand. Can you repeat your first question again?
So occult Hep B, that's like little very specific HBV cohort. These patients basically show very low HBV DNA levels, either in the liver or PBMCs or wherever, but do not have any surface antigen.
No, we didn't analyze them.
Okay. So I was just thinking if they also behave like the recovered patients.
Yeah, this we didn't analyze, but it could be possible that they do because they kind of control the virus, but I cannot give an answer. I mean, regarding my experiments I did. So I don't know.
Okay, thank you.
Thank you.
Thank you for that.